So we know how scary cancer can be and how difficult it can be to treat just because of their survival ability and their proliferation ability as well as their spread. Surgery can be an effective way to treat cancer cells when they haven't spread too much, but when they have spread to a certain extent, that's when we have to start looking at other treatment options such as chemotherapy. In today's video, we'll be taking a closer look at how these type of non-surgical cancer treatment plans can have an effect on the cell cycle control mechanisms and the DNA damage response of cancer cells. To do so, we'll compare a cell cycle of the normal cell versus the cell cycle of a cancer cell. And specifically, we'll be taking a look at the DNA repair mechanisms for both types. A normal cell usually goes through about 40 to 60 rounds of division before it dies. As you can see in the G1 phase, which is the very beginning of the cell cycle, there's a checkpoint before it goes to the next cycle called the S phase. And at this point, the cell checks for any type of DNA damage before proceeding to the next steps. If there is damage in the DNA present, then the cell goes to one of two options. First, it checks if the DNA can be repaired. If it can, it does it by using a tumor suppressor gene called P53, which calls for other enzymes to be activated. Once the DNA is fixed, it can go to the next step, to the S phase, and so on. If the DNA cannot be fixed, then it goes to cell death, which is called apoptosis. The key point here is that a normal cell has to fix any DNA damage before it can continue on to the cell division process. On the other hand, cancer cells tend to divide regardless. Cancer cells do not have a checkpoint that checks for any type of DNA damage like the normal cells do. And as seen in the drawing, these cancer cells continue to divide very closely to each other. Some of the non-surgical treatment options, however, can take advantage of these factors of cancer cells. Radiotherapy and chemotherapy are one of examples that produce direct damage to the DNA in cancer cells. Although in the beginning of this video we stated that normal cells having mutated DNA sequences can later turn into cancer cells, treatment options in chemotherapy can cause way too many unrepaired chromosomal breakages in the DNA of these cancer cells. This will later lead them to being killed during the proliferation process. Another option is to use drugs that directly inhibit the DNA repair pathways that cancer cells use. Many cancer cells don't have the BRCA1 or BRCA2 tumor suppressor genes that are present in normal cells. As a result, they have to use some other repair pathways such as NHEJ in order to maintain DNA stability. This will kill the cancer cells from chromosome dysfragmentation when they're attempting to actually divide. The important idea here for all these treatment plans is that they shouldn't affect normal cells the same way they affect cancer cells, because normal cells have a means of fixing DNA before they begin to reproduce.